Hello students, today we will learn the concept of herbarium. Now this topic comes under taxonomy, you know that, isn't it or not? Now if someone asks you what is herbarium is nothing but a collection of dried specimen, preserved specimen which are used for identification, nomenclature and to study the nature of the plants and given a vegetation in a particular area. It is taxonomically very important because herbarium plays a major role in plant identification. Yes, so for every taxonomic studying student, it is very important to learn the concept of herbarium. So once again I will revise you. Herbarium is nothing but a collection of dried specimen mounted onto a thick paper which is given a label, it is given identification, it is given nomenclature and also it is arranged according to one or other approved system of classification. Now if someone asks you what can be done with the herbarium, we know that plants are of various types. You come from algae, small algae, single celled algae to large trees, isn't it or not? So how to prepare a herbarium? Now in case of small plants like herbs, we usually the herbarium specimen is a complete plant. So a whole plant is taken out, dug out, and a herbarium specimen is made from it. Now in the case of long twiners, they are usually a single branch of nearly 25 centimeters long is collected and it is dried and mounted. Yes. Now if you are taking into consideration plants like certain kinds of xerophytes, especially like succulents which are water storing or mucilage storing plants, then in that case we cannot dry them up. Then what is the other method? In such kind of plants which cannot be dried up, they are usually put in a glass jar filled with formalin solution, which is a preservative. You know that, isn't it or not? In cases of small plants like lichens and different kinds of mosses, usually the plants are air dried and they are preserved in plastic pouches, sealed plastic pouches. And in case of gymnosperms, we know that gymnosperms leaves, which we call them as fronds, and their reproductive parts are quite large. They cannot be preserved as such, isn't it or not? Then what has to be done? They are usually stored in large glass bottles. Yes, this is how the collection is made. Now let's try to see the history of herbarium. Now this word herbarium was first proposed by a taxonomist called Tom Ford. Yes, and the method of drying the plants and preserving them was actually practiced by another great taxonomist called as Luca Guinea. Yes, he is the one who started the Pisa Botanical Garden in Italy. So he is a very prominent taxonomist. Yes. Now at that time, Luca Guinea used to stitch the plants onto a thick leather sheet and preserve them. But the latest concept was introduced by the father of taxonomy, that is Linnaeus. He has started preserving and collecting the herbarium and preserving them on horizontal sheets of papers and arranging them and preserving them horizontally. So these three people get the credit of what we learn about the herbarium concept today. Yes. Now if someone asks you what is the method of how does a herbarium help us? As I told you, herbarium is nothing but a dried specimen. We will try to tell you what is the nature of the plant, whether it is a herb, shrub or a tree, what is its nature, how does it flower. Yes, every nature of the plant, the taxonomic characters can be learned looking into a herbarium sheet. Not only that, this herbarium is a depository of lot of information. Like if you want to learn about the vegetation of a particular area, then the best thing is to look into the herbarium of that local area. Yes, if you want to know the palynological data, embryological data, then this is the one. Yes, any research work, related work, here the herbarium preserved specimen can pave way for learning about what a plant used to be and what is a plant now. Yes, not only that, for a taxonomic student to understand the concept of taxonomy, herbarium sheets, learning them, preparing them is a vital method of learning the subject. Yes, so herbarium preparation, collection, learning the concept is very important for a taxonomy student. Yes, now let's further learn about it. As I told you, herbarium collection is there from past 400 years. Now the world famous herbarium resides in the Royal Botanical Gardens. We know that the Royal Botanical Garden houses large amounts of collection of herbarium, and there are worldwide there are many Taxonomic institutes are there, research institutes are there, local bodies are there, universities are there, which have their own collection of the herbarium, and it is estimated that all these herbarium put together has nearly 250 million amount of specimen are there in different herbariums of the world. Yes, so if you are taking total world into account, Royal Botanical Garden houses the largest collection of herbarium, and if you are looking into India, it is the Botanical Survey of India which has its headquarters at Calcutta, which houses all the major large amount of Indian specimen over there. Yes, now the concept of herbarium, the first herbarium botanical institute was set up by Linnaeus himself in Sweden. If we are talking about India, yes, the concept of herbarium was introduced by the famous taxonomist Roxburgh. Okay, so this is all about a bit of introduction regarding the herbarium, its prominence, etc., etc. Now let's look into what kinds of tools and equipments that we need to prepare a herbarium. Yes, I always told you taxonomy is something that cannot be learned in a classroom. It is a field study. Yes, now let's look into the different kinds of tools that we require when we go for herbarium preparation. Yes, so what we need, let's just briefly look at them. We need a field press. We need a vasculum. Yes, we need a notebook. Yes, we need secateurs. We need a small sickle also. It can be a shear. We may also need scissors. Yes, we also may need scissors and hand lens. These are the few basic equipments that any taxonomic student will go into the field to collect the herbarium specimen. Yes, so a field press is a metallic framed structure or a wooden framed structure which is used to press the herbarium then and there in the field. Yes, and a vasculum, if it is not possible for the herbarium specimen to be pressed in the field, then it is collected and preserved in an airtight container called as vasculum, which is usually made up of tin or aluminium. Then a notebook is the primer and the most important part when we go into the field, because then and there we need to make a note of it, what is the area and what is the time, what is the season, who has collected it. Everything has to be noted down in the notebook. And remember one more point, when we are going for a herbarium collection, we have to go for herbarium collection throughout the year, 3 seasons or 4 seasons depending upon the country, because a plant which is usually a perennial plant will not be the same each season. So phenological characters all has to be grasped. That's why herbarium collection has to be throughout the seasons. Yes, and then secateurs, sickle, shear or scissors, these are used to cut, prune, take out the specimen, whether it is a herb, shrub or a tree, but from where we have to do. Hand lens is used when we are whatever collecting herbarium specimen is too small to observe. Hand lens will help us. These are the few basic things, and of course don't forget about taking the blotting papers, or if the blotting papers are not available, then you can just take old newspapers with you so that you can immediately press the herbarium specimen over there. Okay, so this is about the tools that we are going to take when we are going into the field. Yes, now let's look into the process of making a herbarium. Yes, it is a stepwise process. The first step is collection. The second step is drying and poisoning. Yes, the third step is mounting. Fourth is labeling, and fifth step is arranging them, nomenclature, that is giving a name to them, and storage. Yes, usually we call it as filing and storage. These are the 5 basic steps that we follow to prepare one single herbarium. Okay, now first part we have just discussed, that is collection. As I told you, whenever we go into a field, usually if it is a small plant, the entire plant is taken out along with the roots for collection. If it is a tree or if it is a long shrub, then usually the most appropriate is we have to collect at least a twig of 25 centimeters in length, and that particular twig should show all the characters of the plant. That is, according to Bentham and Hooker classification, we usually go for the flower morphology. So it should be able to show the stem characters, the leaf characters, the flower, nature of the fruit if possible. All has to be there depicted in this 25 centimeter long twig, and nearly we have to collect 4 to 6 identical specimens from the same plant, so that if one is damaged, another is there for us to recover it and go for the mounting. Okay, this is about the first one. Yes, and also remember that we have to collect in all the 3 seasons depending upon the phenology of the plant. Now when we collect the plant, if it is possible, pressing the herbarium specimen has to be done in the field. Most adequate method is that. Otherwise, if it is not possible, you have to get them back into the laboratory, clean off any external soil surface or any contaminants is there, clean them off, and immediately put them under pressure between the folds of an old newspaper or the blotting paper. Keep weight on it and keep changing them every 24 hours. Now we go into the second step, that is drying and poisoning. As I was just telling you, drying is nothing but taking out the entire moisture from the plant herbarium to keep it completely dry, because any little amount of moisture can completely lead to fungal infection, completely destroying the herbarium specimen. So we don't want to take a chance over there. So drying can be under artificial light, sun drying can be there, or we can put them under shade depending upon the kind of the plant. Yes, after the material has completely dried, for storing them for longer period of time, we need to poison them, so to take out any other contaminants. So for poisoning, usually we use mercuric chloride with ethyl alcohol. Yes, this is the combination which is used for poisoning. Yes, where we dip the dried specimen into it for 2 or 3 minutes, half an hour of time, and then take it out and go for mounting. Yes, but mercuric chloride is a dangerous chemical which is carcinogenic in nature. It is mutagen. That's why it is not that safe to handle it. So now the most safest one is lauryl pentachlorophenol. This chemical is now used for poisoning. Yes, this is the second step. Now let's look into the third step. Once the specimen has dried properly, the next step is to mount. How it is mounted? Mounting is done on a herbarium sheet. So a herbarium sheet is nothing but a very thick handmade sheet, and its size is also quite determined. It should be 29 into 42 centimeters in length, where we have to put the specimen in the middle of the paper, and either we can stitch it or can glue it or put a cellophane tape and mount it onto the herbarium sheet. And plant parts like delicate flowers or fruits, if they get detached, we can collect them in small pouches, plastic pouches, and clip them adjacent to the mounted herbarium specimen. This is all about the mounting. Yes, the next important step is labeling. If we look into a herbarium sheet, if the herbarium specimen is over here in the middle, we find that on the right hand down corner, we usually have a place for labeling. Yes, and what do we label over here? The labeling provides us with all the details of the particular specimen that is mounted onto the herbarium sheet. That is its name, common name, botanical name, collector's name, place, time, region, family to which that particular plant belongs, and other details whichever are required. Yes, all the details are given in labeling, which is quite helpful information for future taxonomic studies or rated studies, or when we go for monograph preparation, anything. This particular labeling is quite vital in herbarium preparation. Yes, the last part is, as I told you, after we label, we need to file and store them. Yes, and how is the filing done? As I told you, Linnaeus was the person who has said that horizontal filing is the most appropriate method of the filing. So all the specimens that belong to a common genus or all the genera belonging to a common family are stored in a horizontal box, and they are arranged one over the other very neatly and labeled outside. Which family members are there, all are labeled and put outside the box. Yes, this is about the filing. Yes, then what about storage? I said that herbarium is a preserved material which is stored for n number of times if taken proper care. Yes, so storage is taken care with regular observation. So usually mothballs, naphthalene balls, and if required DDT sprays are also done periodically, so that any pest or any fungus, if they are remaining over there, can be completely removed, and this particular herbarium sheet can extend its life more. Yes, so this is all about the herbarium concept. Remember that herbarium concept is a very important topic, whether it may be for field study, for a future taxonomist, or from the examination point of view. Along with this YouTube lesson, I am also going to send you another video link of actual herbarium collection that has been done, that can be done from the field, for easy understanding of this concept. Hope you have understood the concept. Thank you.